In this video, we will discuss about lectin-glycan interactions and how this lectin-glycan interactions helps the immune system or the immune cells to detect the pathogens and generate immune response against those pathogens. So there are some siglags that are present within the host siglags and also uh, other lectins such as galectins, galectin 3, galectin 1, and this Siglec 1, Siglec 5, Siglec 7s, all these have the capability to detect the receptors of the pathogens, and they usually detect it using the carbohydrate structures that are present on the pathogens. And by detecting if those Siglecs are those receptors are present on the antigen presenting cells, then they will trigger this intracellular signaling pathways that are mentioned here, such as this galectin 1 will provide these antigens uh, informations of the pathogens to the T cells, and then that will helps to activate the T cells and generate a immune response that is specific to those antigens. And along with the CLEC1, the C-type lectins will also help in determining those pathogens through with the help of the carbohydrate structures that are present on the pathogens, and they will also help in activating T cells through this major histocompatibility complex 1 or 2 that can be seen here. And the other part is that is it can activate the other siglecs can activate this intercellular signaling pathways such as SRC kinase SYK that will help in the upregulation of this NF kappa beta, and there are other receptors that will also upregulate this NF kappa beta, and that will help in the maturation of this antigen presenting cells, and at the same time it will also help in the T cells differentiation and macrophage activations, such as, and the T cell differentiation into is Th1, Th2, and Th17. This is about the antigen presenting cells, uh, how they can help in the immune response against the pathogens through the informations that are present on the in the form of carbohydrate structures on the on the pathogens. Now we will understand uh, how this immune response maintain its own balance. That is, when there is no pathogens are available, how the immune response can suppress itself. So there are several galectins. Galectins that plays that roles, such as this galectin 1, galectin 3. It helps in this immunological synapses, whereas this galectin, when the galectin will interacts, then it will also helps in this regulatory T cells such as T regulatory 1 and CD4 T1 and CD45 T1, and it will upregulate this galectin 1 and galectin 10, and this interleukin this T regulatory 1 will upregulate this interleukin 10. And what will happens when this interleukin 10 gets upregulated, that, and also when this CD4 plus CD25 plus cells are regulated, it will produce more galectin 1 and galectin 10. And this galectin 1 and galectin 10, apart from this, as you know, that T regulatory cells function is to suppress the activated T cells, as there are no, and because of the understanding that there are no more, or the amount of pathogens are reduced within the host. At the same time, this galectin 1 and galectin 9 interactions with these T cells also helps in the a decrease in the population of this Th1 and Th17 cells, and thereby promoting these Th2 cells, which is resistance to galectin 1 and galectin 9. And so this is about the T cells balancing, and also these B cells functioning can also be affected because of the glycosylation, because of the SELEC1 interaction with this glycosylated CD43 that is present on the antigen presenting cells. So when it interacts with this B cells, then it will promote the this proliferation and survival. So B cells will try to accelerate itself, As B cells, we can know that is the primary form of response. And when it comes to the T cells, so what will happen is that the glycosylated receptors that are present on the T cells in the form of CD43, CD45 T cells, and this T cell receptors also, when they interact with these antigen presenting cells, then along with that, when the Galectins, such as this Galectin 1, 2, 3, 9, interact with these T cells, then it will trigger apoptosis and also this interleukin 10 upregulations. Interleukin 10 upregulations are upregulated only not only because of these T cells interacting with this Galectin 3, but also when this T regulatory T when the regulatory T cells are produced. So, and this will activate, and this will cause this downregulation or control downregulation of the activated T cells population, and thereby act as a anti-inflammatory responses. So these are the balances that needs to be made within the immune response, so that whenever the pathogens are detected, then pro-inflammatory response are triggered, that is shown in the left hand side, and when the uh, pathogens are amount is reduced or eliminated, then anti-inflammatory responses are triggered. It is shown on the right hand side. This is how the lectins plays a role in this immune response. And if you need to understand more about this lectin-glycan interactions in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.